Good afternoon. Welcome to the Nanomedicine session. Our invited speaker of today is James Moon. James Moon is from the University of Michigan. He studied bioengineering at Berkeley, and then he did his PhD at Rice University, followed by a postdoc in materials science and biological engineering at MIT. And currently, he and his team work on advancing cancer immunotherapy and vaccine design by applying engineering and pharmaceutical tools. Today, he'll talk to us about engineered biomaterials for cancer immunotherapy.
Okay. Hello, everyone. Thank you very much for the organizers for this kind invitation to come here and share our latest research. We are working at the interface between pharmaceutics and immunology, and we are trying to improve the patient outcomes in the cancer immunotherapy area, to be specific. And we have some cool nanotechnologies that we want to share with some promising preclinical outcomes. And based on the research that I'm about to describe, we started a new startup company called Evoke. So that's my disclosure slide. So there's a lot of talk on cancer immunotherapy, voted as the scientific breakthrough of the year in 2013 by Journal Science. This field has been galvanizing the whole field of oncology. And the focus of all this is on immune checkpoint blockers. These are antibodies that allow existing T cells to come and basically allow tumor cells to get killed. Not sure why it's in auto advance. So these are antibodies that come in between T cells and tumor cells and basically allow T cells to come and then kill their target tumor cells displaying antigen of interest. And whole field of the cancer immunotherapy is provided an overview is shown here. Basically, when tumor cells are dying through the process of necropsis of apoptosis, they release antigens, and they are picked up by immune cells, such as dendritic cells, and that trigger cytotoxic T cells that try to go into the tumor microenvironment and kill their target tumor cells. But tumor cells have a lot of immunosuppressive environment, different cytokines and proteins, to block the interaction between T cells and tumor cells. And this is when these monoclonal antibodies, immune checkpoint blockers such as Keytruda and Opdivo, come in, allowing existing T cells to kill their target cells. And it's making striking clinical success in multiple indications. And this is a case of advanced melanoma patients who are not responsive to chemo or previous radiotherapy. And what you see is that with the monoclonal antibody, up to 20 to 30% of patients are long-term survivors without signs of relapse. And this is a fantastic news for cancer patients, but it still shows these 70% of patients that are not responding well to these monoclonal antibodies. And that shows the major the obstacles in the field, which is that when patients don't have enough number of T cells to begin with within the tumor, there is no target for antibodies to work against. They don't have enough T cells in the tumor environment, called as cold tumors. And we think that's one of the major reasons why immune checkpoint blockers are not working in 100% of cases. So what we are doing is that we are trying to work on the upstream process where we try to use a vaccination approach or chemotherapeutic approach to increase number of T cells that can recognize tumor. And then when we combine with immune checkpoint blockers, hopefully we'll have much higher patient response rates. Another interesting idea that's coming up in the field of cancer immunotherapy and genomics is the idea that a lot of cancer patients have mutations that are specific to that patients. So if you look at each individual cancer types on the x-axis, and the y-axis shows non-synonymous mutations that's found on cancer DNA. And what you see is that subsets of cancer, such as lung and melanoma, have high rates of mutation, up to 200 mutations found in tumor DNA. And that's probably because in the case of lung cancer, smoking, and melanoma due to UV radiation that promotes mutations in the tumor genome. And what the research in the field has shown that every cancer patient have different mutations, and when these mutated protein gets chopped into fragments and presented on the surface of tumor cells, the peptide itself is very different from one patient to another. And that takes us to this idea of using personalized cancer vaccination to promote cancer immunotherapy, to synergize with cancer immunotherapy. Basically, as I described it before, cancer cells have mutations that are unique to that cell type, while healthy cells don't have that mutation. And these fragments that are displayed on the surface of cancer cells can be detected by T cells, CD8 or CD4 T cells that can go and kill their target tumor cells. And this field is really taking off. Only 3 years ago, there were papers showing their proof of concept in preclinical studies, where in murine tumor, you take tumors out, do DNA RNA sequencing to find these epitopes called the neoantigens, and then you do vaccination to show efficacy. But there have been a couple of seminal papers that just came out last year where they used a peptide or mRNA-based approaches to go after this mutated epitopes. And in my lab, we are working on the peptide aspect of a cancer vaccination. And we try to come up with a better delivery technologies to promote much stronger cytotoxic T cell responses. So this slide shows the overall obstacle in the field of cancer vaccination, especially on peptide-based systems. What happens after subcutaneous or intramuscular injection of peptides or small molecules is that a lot of it gets washed away into systemic circulation, but you want your vaccines to go to draining lymphoid tissues where there are a lot of high frequency of immune cells. So if you inject small molecules, small vaccines, small molecular weight vaccines, you see very weak T cell responses to this delivery problem. And what we found out was that if you have a nanoparticle system, such as synthetic high-density lipoprotein that we are using in the lab, after sub-Q injection, what we found out was they drain very effectively through the lymphatic vessels, carrying whatever is attached or loaded into the core directly to lymphatic tissues to draining lymph nodes where they are taken up by dendritic cells, and you get very strong T cell responses. So just to speak briefly about our nanoparticle system, this is maybe very familiar with you from the ongoing work on HDL in the institute. We are using synthetic high-density lipoprotein that's composed of just a phospholipid and peptide, very simple to manufacture. And this actually went through phase 1 clinical trials for cardiovascular applications to use HDL as ways to lower bad cholesterol in patients. So we have experience with manufacturing and phase 1 clinical experience, and they have been shown to be very safe after IV infusion in cardiovascular patients. But what we are proposing is to use this as a new tool to deliver vaccines, and we are proposing it to use a sub-Q route of delivery with orders of magnitude less amount of drug that were used in clinical trials for cardiovascular applications. A little bit about formulation. We start out with a standard method of making liposomes, where we dry down lipids, hydrate them in the presence of aqueous solvent, and then in the presence of a 22A peptide that has alpha helical structures, as found in apolipoprotein, they self-assemble into very tiny 10 nanometer nanodisc-sized shapes. And we went through a series of optimizations in terms of lipids, and then peptide fragment sequences. And what we arrived at was a formulation that's very stable, shown here. After 1 month of storage at 4 degrees in PBS, they are very stable. And we are taking that to deliver peptide and adjuvant fragments. And here, this is showing how we attach peptides into the system. We have a functionalized lipid that can readily react with any thiol-displaying antigens. And we have worked with antigens, cysteine antigens in the core, or we just, for the case without the cysteine antigen, we add it at the N-terminus. And we can work with any peptide length between minimum epitope length of 8 mer all the way to 30 mer peptides. And typically, peptide cancer vaccine clinical trials are done using 8 to 30 mer peptides. That's what we're using. And for the adjuvants, we need a strong immunostimulatory adjuvants to stimulate immune responses. Because without adjuvants, this peptide alone is not going to trigger immune responses. And for that, we use a DNA-based adjuvant called CpG, where we tether cholesterol onto CpG. And we chose cholesterol because HDL is a natural carrier for cholesterol. And by simple admixing mixture of the two, we see very nice loading of the cholesterol CpG into the system, up to 98%. So what you have at the end is a nanodisk system that's co-loaded with antigen and adjuvant that are very homogeneous at 10 nanometers. And we can readily store filter it at 0.22 micron, store frozen for many months. And then if you thaw right before use, they still maintain their original shape and size. So this is the big picture that we have. The vision is that we would make kilograms of synthetic HDL nanoparticles, which we know from our previous manufacturing of clinical materials. And then we would aliquot them and freeze them as individual aliquots. And when patients' DNA, RNA are sequenced and new antigens are identified, you can take a couple of vials out, mixed with the new antigens, and put with adjuvants. And now you have a personalized vial of cancer vaccine that's ready to go. And how do they work in vivo? So first thing we did was we injected our nanoparticles tethered with a FITC conjugated antigen, a subcutaneous base. And then we look at draining inguinal lymph nodes after 24 hours. And this is how much antigen you can deliver with our nanodisk system. That's about 28-fold better than injection of free peptides. And as I mentioned before, injection of free peptides or small molecules sub-Q results in systemic dissemination of vaccine, which is known to cause immune tolerance. So by simply tethering the same amount of antigen onto the nanodisk, we see a dramatic increase in antigen delivery to the right organs. And that results in a very striking level of T-cell responses. So here we put one of the cancer neoantigens from colon carcinoma, a single epitope into the nanoparticle system, and then we did the vaccinations 3 times in mice. And shown here is on the y-axis the frequency of T cells in blood, mouse blood, that are specific to this single epitope after 3 rounds of vaccination. And what you see is that if you look at the nanodisk shown on the blue bar, up to a quarter of all CD8 T cells in mouse blood are single epitope specific against this tumor antigen. And that is about 30-fold better than the standard adjuvants that are in clinical trials, including free peptide, CpG mixture, or free peptide CpG emulsified with montanide. This is oil in water emulsion, also known as an IFA, very potent adjuvant in phase 1 clinical trials. So at the end of the day, what you have is a very strong delivery system that enhances a delivery of antigen and adjuvant, and that results in very strong level of T-cell responses. So here we proceeded to test their efficacy in a mouse model of colon carcinoma, where we have sub-Q flank tumor. And when tumors are about 5 to 6 millimeters, we initiate the treatments on day 10, 17, and 24 using our vaccine. And what we see here in the blue line is that the nanoparticle immunized mice have extended survival and reduced tumor growth. Whereas the free peptide, CpG, the way conventional vaccines are given, you don't see much efficacy. So this is one of the first demonstrations where we use nanoparticles that are personalized to that patients, in this case mouse, neoantigens targeted against that cell line's mutation. And what's really interesting is when we combine this with immune checkpoint blockers, as I mentioned in the intro, If you use the same tumor model and then initiate treatments on day 10 in combination with immune checkpoint blocker anti-PD-1, now we see up to 85% of mice are eradicating tumors away, creating this long-term survivors. But if you give the conventional vaccines with the free peptide in combination with anti-PD-1, only 30% of mice are responding, as I mentioned in the intro. And another benefit of using immunotherapy is you can now have long-term immune responses against the tumor recurrence. So if you take the survivors from here and then inject the tumor cells again on the contralateral side or IV route, what you see is these mice are 100% protected against the tumor recurrence. So this suggests that you can not only eliminate primary tumor, but also prevent a future occurrence of tumor relapse. So this looks very promising. Also, that was the case with a single epitope mutation, but in the clinical settings, we would go after multiple clones. So here we used a much more challenging model of B16F10. This is melanoma model, where we put 3 different antigens, CD4 and CD8 epitopes, in a cocktail approach. And once again, when we combine with immune checkpoint blockers, anti-PD-1 and CTLA-4, we see 90% of mice are eradicating tumors away. Whereas the conventional combo therapy with a soluble vaccine, you see about 40% of 30 to 40% mice are responding. And once again, you have this long-term immune responses against tumor relapse as well. So that's very exciting results. And what we are also interested in is using this as shared antigens as well. So far I've talked to you about using personalized approach to have a vaccine that's tailored to everyone's mutation. But there is also a lot of research ongoing on shared antigens, such as HPV-16 infection, where E7 antigen is known as a viral antigen. So in this model, basically, where we have a TC1 tumor cells, these are epithelial carcinoma cells that have HPV E7 expression. And after IV injection, they get stuck in the lung capillaries to have a lung metastasis. And without treatments, the mice succumb to tumor within about 30 days. But with our nanodisc immunization given by sub-Q or intranasal route, we see nice eradication of the tumor mass from lung tissues in the majority of cases. And we think that's all driven by this strong anti-tumor T-cell responses, as shown here. This is showing the frequency of T cells in blood that are tumor specific, in this case, this viral antigen specific, and up to 30% of all CD8s in the nanodisc immunized group are tumor specific. And HPV infection is also implicated in head and neck cancer as well as cervical cancer. So we use the same tumor model in an orthotopping model where we implant CT1 cells under the lip to have a model for head and neck. And what we see is the nanodisc immunized mice are eradicating tumors, 100% cases. With the intranasal route, you see some reduced efficacy, but that's still better than giving free peptide vaccination. And in an orthotopic model for cervical cancer, we also see that. This is much more challenging distant tumor model, where the nanodisc is eradicating tumors in about 40% of mice, whereas the intranasal group is not working as well. And we are trying to work out the mechanisms of this, but what we think is going on is that initially the tumor cells are elicited within the circulation shown here, but they are slowly penetrating into the lung tissues or wherever the tumors are, and they are also penetrating all the way into the vaginal tissues in this mouse model of cervical cancer. So far, we have shown the efficacy of the nanodisc system, either using personalized new antigens or shared antigens in multiple murine tumor models. And we started a company, as I mentioned before, called Evoke Therapeutics, that's trying to put this into the clinic. In Michigan, University of Michigan, where I'm from, majority of cancer patients are entered into our program, and their DNA is sequenced. And the database is available to mine the neoantigens in the system. So we think we can readily implement this into the whole molecular tumor boat and then implant this into the phase 1 clinical trials. And we are also collaborating with Dr. Nina Bawaji, whose lab is like 5th floor in this building, who is also pioneer in neoantigen vaccination. She is actually treating patients with the neoantigen peptides, and we are initiating collaborations to try this in patients here as well. So we'll see how that goes. Switching gears a little bit for the remainder of my talk, I want to also touch briefly on the idea of combination chemo immunotherapy. So this is a relatively new area of research where people are seeing that certain chemotherapeutic agents can cause what's called immunogenic cell death among tumor cells, such as doxorubicin and oxaliplatin. When they are used to treat tumor cells, they not only kill tumor cells, but also kill them in a way to trigger immune responses by releasing what's called the danger signals, such as HMGB1 or calreticulin on tumor cell surface. So the end result is that the local immune cells will come in, engulf the tumor cells, and then trigger T cell immune responses against this tumor in a process called immunogenic cell death. So what we wondered was, since HDL, this system that I just described, has been tried in hundreds of phase 1 patients in phase 1 clinical trials by IV route, can we use it to deliver this known inducer of immunogenic cell death, doxorubicin in this case? So here we basically utilized this previous research where we have implanted a xenograft tumor into mice. And after IV infusion of HDL, they go straight into the tumor, probably through cholesterol efflux mechanism. And we previously delivered different chemotherapy agents into the system. So based on this, we came up with a hypothesis that if we attach doxorubicin into the HDL and then give them by IV route, will they go into the tumor, accumulate the drug, causing them to undergo immunogenic cell death. And when we combine with the immune checkpoint blocker, will that result in much better efficacy? And this is how we approached this. We used a pH-sensitive linker that allows embedding of doxorubicin into the lipid layers of HDL. And this pH-sensitive linker is cleaved under the pH of 5, pH found in endolysosomal conditions of tumor cells. So in pH of 7.4, after IV infusion, the nanodisc would be very stable, holding onto the drug. But within pH of 5, within intracellular compartments, they would release the drug and killing them. And indeed, if you treat tumor cells, colon carcinoma cells with our nanodisc, you see nice killing, slightly reduced efficacy than free peptide, free drug, doxorubicin, but you still see good killing. But what's interesting is you see increased elicitation of this immunogenic cell death. HMGB1, the marker of immunogenic cell death, goes up after the nanodisc treatments. And after, so here we implanted CT26 cells sub-Q and then give them either IV infusion of doxorubicin or HDL carrying nanodisc. And what we show is a good efficacy of the doxorubicin loaded into the nanodisc, where we see extended animal survival. And a lot of people have done this type of work before in the nanomedicine field, so that was not so surprising that our nanodisc is exerting stronger anti-tumor efficacy compared with free drug. But what was interesting was what happens to anti-tumor T cell responses. So after nanodisc doxorubicin delivery, we see dramatic increase in anti-tumor T cells that can recognize this CT26 cells. And what was even more interesting was when we combined this with anti-PD-1. When we use the dual co-therapy, we see even dramatic increase, and up to 17% of all CD8 T cells in mouse were tumor specific, and that resulted in much better efficacy. So here, the same colon carcinoma model where we implant the tumor sub-Q and treat them on starting day 8. What we see is the nanodisc plus anti-PD-1 showed nice eradication in the majority of animals. But if you give free doxorubicin chemo drug plus anti-PD-1, we see a progression. And in fact, up to 85% of mice in the nanodisc co-treatment group had eradicated tumors away, whereas if you have a free dox plus anti-PD-1, only 3 out of 8 mice showed a tumor regression. So this is very exciting results showing the promise of nano-mediated delivery of chemo in combination with the immune checkpoint blockers to have anti-tumor efficacy. And as I shown in the first part, you can also have long-term immune responses against tumor rechallenge. So these survivors were 100% protected against tumor recurrence as well, showing their long-term anti-tumor memory responses. And we also tested this in a much more challenging orthotopic tumor model where we implanted the same cell line expressing luciferase into the cecum to have a colon tumor model. And this kills mice within 20-30 days, as shown in this luciferase imaging. But with anti-PD-1, this is resistant to, this cell line is resistant to anti-PD-1, so you don't see much increase in efficacy. And with a combination with free dox and anti-PD-1, you see some mice are responding, but not majority. But with nanodisc immunized mice, even in the distant tumors, you see nice eradication in, or much more reduced luciferase signal, and that's shown here, night and day difference between the other formulations. And what we also observed was a very strong elimination or reduction of signal in the liver metastasis. These tumor cells metastasize from colon to liver. So if you look at the liver tissue, the nanodisc in combination with anti-PD-1 is significantly reducing liver mets, and that's translating to up to 85% long-term survivors with the nanodisc combination group, whereas other formulations didn't have this efficacy. So in combination, what we show is by tethering the old chemotherapy drug, Doxorubicin, into the nanodisc and doing IV infusion, we see much better accumulation of the drug within tumor cells. And these tumor cells undergo this immunogenic cell death that can synergize with the immune checkpoint blockers to exert very strong anti-tumor efficacy. So I'll just quickly go through the third part since we want to leave some time for Q&A. Another concept that we are pursuing in the lab is using gold nanoparticles for combination immunotherapy. A lot of people have used different inorganic materials to basically ablate tumors away. So for example, if you inject gold nanoparticles with a certain shape and size and then shine light, these particles will absorb light and then convert that to heat, basically cooking our tumor cells away. And what we found out was that if you combine them with a specific concentration of doxorubicin, that can also trigger this immunogenic cell death that I just described, triggering strong anti-tumor T-cell responses. So just quickly, what we show is that if you inject these nanoparticles into the tumor and then shine light, they basically eliminate tumors away. And then they trigger this anti-tumor T-cell responses that can be detected in the systemic circulation. And when you take the survivors and then challenge them using the tumor cells again, they basically eradicate prevent a tumor engraftment. So a lot of people have used this what's called the photothermal therapy, using light to trigger photoablation of tumor cells. Local tumors, and that has been the major limitation because you need to shine light into this local tumor with the golden nanoparticles to eliminate tumors. But what's limiting the field is what happens to the distant, small metastases that are hard to reach using conventional light. And when you have this strong anti-tumor efficacy in systemic circulation, you can now think about eliminating distant tumors. And that's what we tested in this tumor model, where we have 2 tumors in mice and then treat only one side using this photothermal therapy and see what happens to the distant, untreated tumors. And this has not been done before in the field. And what we have shown is that the primary tumor, in combination with the photothermal therapy and dox, is completely eliminating tumors away from local tissue. That's not unexpected. But what happens to distant tumor is very interesting. So in untreated distal tumors, this combination with chemo plus photothermal therapy, up to 85% of mice are eradicating tumors away. Whereas if you have just photothermal therapy, you don't see much efficacy. Free chemotherapy, you don't see efficacy as well. So that resulted in good survival benefits in these mice. And we have also shown that this is mediated by CD8 Tcells. So you basically ablate CD8 Tcells during the treatments, and you lose this survival benefits. And if you deplete NK cells, you also see decreased survival benefits. So we showed that this is a CD8 and NK-mediated ablation of the distant tumors. And we have also shown this in a orthotopic model where we implant the TC1 head and neck model into the lip and then inject the tumor cells IV to have a lung metastasis. And you basically treat only one side, the head and neck area, and see what happens to the lung tissues. And in the local head and neck model where we inject gold nanoparticles and doxorubicin together with the light, you see nice eradication of these local tumors. But in the distant site, what you see is the nanoparticle plus doxorubicin is eradicating, well, significantly reducing the tumor-associated signal by order of magnitude compared with other formulations, showing that this local treatments results in reduction in tumor mass in a distant sites that are not treated. So we think this has good implications for ongoing clinical trials. Basically, there is a clinical trial that's ongoing where photothermal therapy is used to treat head and neck cancer model, but it's not able to treat distant tumors, and we think this may have a place in that clinical setting. So overall, I want to thank lab members who have carried out all this fantastic work, especially Dr. Ju-Tek Nam, Sejin Son, and Rui Kwai. And I want to thank our funding sources, as well as our collaborators, including Dr. Nina Bawaji from Mount Sinai. All right, thank you.
Thank you for this very interesting talk. I see there's a couple of questions. Go ahead. Nope.
Electrical engineering 101.
Oh, yeah, it does. Yeah. Okay, I'm a chemist. Okay, leave me alone. Hey, so very nice, and thanks for this talk. So what's your strategy? Are you thinking, so I'm thinking about your company, right, and translating this to patients. So you'll have to get approval for every new compound that has a different antigen or peptide. So, when you talk about this personalized way of doing this, I can imagine it will work if you have a general antigen that works, for a specific tumor subset or something like this.
Right.
But so, could you comment on that?
Yes, so that's a big question, and FDA is catching up. There are a lot of companies that are trying to use new antigen or personalized antigen cancer vaccines. So what the guideline so far is that once you go through a couple of patients and show the quality control and characterizations of your vaccine formulation that FDA is happy with, they would let the subsequent patients treated using the same procedure of manufacturing, so that the release of materials is not too delayed.
Yeah, but so the company is in anticipation that you will be allowed to do this.
Yes, there have been free peptide clinical trials using free peptides that have implanted that in phase 1 clinical trials.
Okay, so you do subcutaneous injections, right?
Yes.
So do you do that in a foot pad, or do you do this, or around the tumor?
Around the tail base.
Okay,
in this model,
okay.
but or head behind the neck.
Yeah, and then last question. So you showed that you have more efficient delivery as compared to the let's say the bare antigen. So what was labeled?
It was OVA albumin synthetic peptide, widely used peptide,
Yeah, I know.
with a FITC.
Okay, so
And
you
that was
s-
tethered into the nanoparticle system
Yeah.
or injected as free antigen.
All right, thank you.
Hi, very nice work. You showed some of the data with melanoma, and one of the part that melanoma makes it worse in terms of prognosis is brain metastasis.
Mhm.
So I was wondering whether you have worked on trying to deliver
Yes.
across the brain this your
Yes.
compound.
Yes, we are trying that now in combination with radiation therapy. We have some pilot data, but we are not ready to share it yet. But it's looking promising, and we are also working with a glioblastoma model as well. So hopefully we'll have enough number of T cells in circulation that can penetrate blood-brain barrier. How much does it cost for patient? I think I heard that. Yeah, it's pretty expensive. We are expecting about 100,000 per patient. But if you consider immunotherapy right now, immune checkpoint blocker, that's about $100,000 per year. And we anticipate maybe this will just require one round of treatments. Yes. So that's something we need to collaborate with other people, including Nina Bawaji. And they have a very robust genomic prediction tool, how to predict which antigens to go after. Because I showed in the beginning, each tumor cells have up to several hundred mutations. So there's a whole prediction tool of which peptide is, which protein is expressed and high amount, how they are degraded within intracellular compartments, and how they are loaded onto the surface with MHC1 or 2. So there's a whole line of prediction that needs to be done. And maybe AI can be implemented if we have enough data sets to train them as well. Yeah.
I've got a question too.
Yes.
I was also intrigued by the subcutaneous injection of the HDL particles, because we use those too, and then we always inject intravenously. So I was wondering how much, do you know how much of your particles end up in the lymph nodes?
We are characterizing them, but we think we are seeing about only 5% or less. And so there's much room for improvement, but even that is 30 times better than free peptide vaccination. So that was a striking result, but that is something we need to improve upon. Yeah.
And compared to intravenous injection?
Compared to intravenous, we haven't done. It's very hard to reach lymph nodes with the IV injection. It is going to be very, very low, 0.1% or less. Yeah.
Okay.
All right. Thank you. Okay. All right. Thank you.
You're welcome.